building off of the physiology and the etiology that Dr. Holt put together for us, both Dr. Speidel and Dr. Buchanan have done considerable work with applying the heart scoring system at Harvest. And Dr. Speidel is going to talk to us about a project he has going together with ABS and Cactus Feeders, looking at some of this. Dr. Speidel also works at CSU. He's well, honestly, we wouldn't make as much progress in this area without Dr. Speidel. He's a key player in our breeding genetics group. I've known Dr. Speidel since before I was at CSU, but he's been in a professorial role there for the last 14 years. And so, Scott, I'm going to turn it over to you. I will point out that both Dr. Speidel and I agree that following Dr. Holt as a speaker is the kiss of death. So, yeah.
Right. There's a reason when you go to a concert, they put the headliner last and they put the opening acts first. So thanks, Dr. Enns. So what I was asked to do is talk a little bit about some of the work that we're seeing with regard to this project that we have funded. Dr. Holt just went through a lot of the physiology, so I'm going to skip through a lot of these early slides pretty quickly anyways, in order to sort of make more time for the end. So again, Dr. Holt mentioned there's a lot of different people who have touched this, and really he's the one driving a lot of this work. But, I'm just kind of put together a list of how many people have touched either our high altitude work or this, feedlot heart failure work. And, we've got numerous faculty members. We've got a whole list of graduate students. We've got health, DVM, PhDs. Some of those are medical doctors from UC Denver. We've got veterinarians, Tim Holt, Frank Gehry, Greta Kraftzer, and then a number of our collaborators between ABS and Cactus, High Plains Feed Yard, Meat Animal Research Center. There's a number of people that have put their time and effort into getting this work to where it is today. So I just wanted to, take a little minute and acknowledge all of their contributions. Dr. Holt was talking about hypoxia. I've got my grad student Isabella, who I listed on the previous slide. She's really the one driving this, driving this project. And she put together this slide at her master's defense, and I really found it a little bit kind of interesting. If you think about cattle, and they're really sort of at a biological disadvantage with regard to their cardiopulmonary system in terms of the size of their hearts and the size of their lungs. And so I don't know where she dug these statistics up, but I'm guessing they're right. I'm hoping they're right anyways. I didn't question her on them. But, if you think about just volume, and she's got right there, the equine lungs can be filled up to 55 liters, and which is pretty impressive. And cattle have 12 liters of air capacity and humans have 6. So, you think about humans, we're really only half of what cattle are. And if you think about the body size, I mean, some of us, not all of us, but if you think about the body size, that's pretty impressive. And so if you look at 12 versus 55 liters, right, what does that look like? And so you can go on the internet and you can find all these pictures of, students and their science classes filling up lungs and showing how big those are. And so you've got a couple of kids there inflating a set of cattle lungs. And then if you pull together equine lungs, look at the difference, right? It's amazing the difference in the size of these lungs compared to the cattle. And so that just kind of, it just kind of reiterates that thought process of, cattle suffering from pulmonary hypertension resulting from hypoxia, right? It's the amount of the capacity of the lungs is much, I guess, reduced, for lack of a better word, with regard to some of the other livestock species. So Dr. Holt went through all this. I'm not gonna go through this again, but really what we're trying to do here is determine the relationship. Which parts of these are genetic? Which parts of these are environmental? Right? And so that's really the key. And the key point here is really starting with that alveolar hypoxia and how they respond, right? And like we said, some of them can have high pressures and still be functional. Some of them are hyper responders. Some of them are, some of them kind of sit somewhere in the middle. So how much of this is genetic? How much of this is environmental? And really, how does this relate to other performance traits or performance characteristics that we routinely measure for their economic relevance? So again, just kind of differentiating, we've got a lot of the same slides here. So differentiating between high altitude disease and feedlot heart disease, really the cause, once that cascade begins, the end result is the same. So what we're trying to do is really understand why some can survive it or tolerate it and some can't. And with high altitude disease, we've got a number of different tools that have been published by breed associations. There's a whole set of guidelines for pulmonary arterial pressure that's published through the Beef Improvement Federation, and pulmonary arterial pressure EPDs. The American Angus Association is publishing a PAP EPD, and I believe IGS is published now as well. So, there are selection tools that producers are at elevation have been using to really, I don't agree with this last bullet point, really producing progeny with lower PAP scores, but what we're trying to do is we're trying to identify those that are more tolerant of those altitudes or more resistant to high altitude disease. So like I said, with feedlot heart disease, our direct cause is really unknown, but these individuals are experiencing the same symptoms as what we're seeing in high altitude disease. So now what does this look like with performance, all right? So I'm kind of taking a step back and looking at, some of the trends that we've seen with our high altitude work, okay? So in terms of our high altitude work, at CSU, we've put a lot of effort into understanding relationships between PAP and the PAP EPD and the genetics associated with pulmonary arterial pressure and our normal economically relevant traits that receive selection pressure. So I've just kind of summarized these in a couple of bullet points, but if you think about this, carcass traits tend to have weak to moderate correlation with yearling PAP. And so when I say yearling PAP, the yearling at a year of age is when breeding or replacement breeding animals are typically measured for their PAP, and that's the EPD that is produced. So carcass traits tend to have a weak moderate correlation with yearling PAP. Heavily muscled cattle may be more prone to risk of higher pulmonary hypertension. In terms of feed efficiency and intake, we tend to see that these high PAP cattle are a little bit less efficient in terms of their performance, feed efficiency. And, I've got average daily dry matter intake in there, but that could be any sort of, feed to gain ratio or anything like that. It's not necessarily average daily intake, but it might be that relationship between gain and intake and everything else. So, and then stress, we've got some papers out that talk about stress and meat quality and relationship with PAP and storage life of product. So it's, there is a relationship between cardiovascular health and these performance traits. So again, here's the scores, or here's all the scores that we are using in this study, ranging from a 1 to 5. So just to reiterate, the 1 over here up on the top left is a normal heart, ranging all the way down to a 5 on that bottom right. And remember, anything scores of 1 and 2 were considered versions of normal, right? And then, or versions of healthy, and then 3s, 4s, and 5s are considered, I'm just calling them affected in my presentation here. So I'm going to break out my results or Isabella's results. I call them my results, but I'm going to break out Isabella's results into 2 pieces: healthy versus unhealthy, and then by individual score. And I think we see some of the big changes and nuances that are happening with each of the individual scores. The stage is up here creeping. I hope it doesn't fall down on me. Okay, so the project, the data that we're talking about is a grant that we received from the Foundation for Food and Animal Research's International Consortium for Antimicrobial Stewardship and Agriculture, ICASA. But there's a number of collaborators on it, CSU, ABS, Cactus Research, High Plains Feed Yard, Veterinary Research and Consulting Services. And really the objectives, you know, we had 4 main objectives, but the big goals for this project was to, 1, determine the relationship between pulmonary arterial pressure and heart score, right? That was one of the big objectives. Examining the role of the underlying genetic component of the disease incidence, determine the effect of heart remodeling on carcass and performance, and then ultimately, if heritable, the development of a selection tool for feedlot heart disease resistance in the term of an EPD. So the first set of data that we're going to talk about is really phenotypic differences, right? So raw phenotypic relationships between heart score and performance traits. Okay, so just to describe the population, so this is a 3-year project. We had just completed year 2, not quite finished compiling all the data for year 2. We're getting ready to start year 3, but at this point, when all this was put together, I'm summarizing the first year's worth of data. And what we're seeing in preliminary results so far, with the addition of year number 2 data, we're following the same sort of trends that we're going to see here. So not much has changed in terms of trends, but we're focusing on beef breeds only. So these are going to be Angus influenced breeds that were raised and fed in the panhandle of Texas. We do have a piece of this that's looking at beef on dairy, so that cross, but most of that data has been collected in year 2, so we don't really have any of that summarized here for you yet. So maybe you'll come to next year's BIF and we'll have some more information on that for you. But so to this point, we've got 1,400 head of Angus influenced breeds, about an equal mix of steers and heifers, and with an average age of about 483 days. So this is just a slide of summary statistics, and I don't have all of the traits that we have on here. I have some of what I think are the important traits. So we've got heart scores, we've got means, standard deviation, minimums, maximums, and there's a lot of information on here. The one thing that you'll notice here is with PAP, there's a big discrepancy in the number of PAP measures, and so Tim had PAP some late in the feeding period PAP measures on a number of animals prior to the start of this project. And then once we started this project, we wanted to see how PAP changed across the feeding period. So we were PAPing animals early as they enter the feed yard at around 9 months of age, and then right before they were harvested. So there's that 178, there's a group of those that have both early and late PAPs. And I'm going to talk about how that progression of PAP changes and what it looks like with heart score from all of these different on that set of animals. Okay. So in terms of the frequency of heart scores that we're observing, all right, so this is just a histogram. We've got the scores over on the bottom. These are all collected in the packing plant, but 1s, 2s, 3s, and 4s. So the majority of what we see are still 1s and 2s. But we're, alarming, well, at least I think it's alarmingly, we've got 29%. So almost a third of them have some sort of affected heart that we're observing at harvest. Interestingly enough, we have no 5s in that, so we haven't observed. I think there were a couple of 5s on the beef on dairy animals that we collected in Kansas, which is not included in this. But right away when they opened it was kind of amazing. When they opened those up, it was just a big wash of fluid out, and those carcasses were condemned almost immediately upon seeing that. So. Okay, so getting on to some results. So this is just a simple mean separation test. So what we've done here is we've taken on the left column, we've got the healthy hearts. So these are the scores of 1 and 2 versus the unhealthy hearts, so scores 3 and plus. And so we just have the different means between the different groups. So you'll see right here. I've highlighted the ones in yellow that are either statistically significant or trending significance, meaning we've got a 0.1 or a 0.08. But in terms of 9-month PAP, this first year, this was a pretty uniform group of PAP measures. It was kind of impressive to see how close the range, or how tight the range was in those PAP measures. But once they got up to 14 months of age, we see that there's definitely an increase in the mean PAP score. And keep this in mind, because when I get to the repeated PAPs, you'll see why this becomes, I think, even more impressive when I look at individual hearts. So we're seeing increasing in PAP as they go from healthy to unhealthy, which Dr. Holt said they would. And so we got what we expected. So good for us. In terms of marbling, we see decreases in marbling score. So as we go from healthy to unhealthy, marbling decreases. As we go from healthy to unhealthy, carcass weight is increasing, but just put a little hashtag next to that, because I'll show you that that's not exactly what we're seeing. There's a little bit of explanation that goes on that when we look at individual heart score. And then if we look at down here, feed conversion ratio, these animals, as they go from healthy to unhealthy, they're becoming more inefficient, okay? So just quickly, I'm going to go through this because I pulled out of these 3 that I highlighted that are significant. So again, the rows that I highlighted here are significant. Here we're looking at heart score, individual heart scores, and their average, or the average PAP, marbling score, or carcass weight for each heart score. So the 3 significant ones I put on graphs because I think it's a little more visually stimulating when you look at it in a graph than you do looking at it in a table. So just to kind of give you an idea, if we look at, so these are least square means. So these aren't raw phenotypes or anything. So these are the predicted averages for each group. If we look at 9-month PAP, we see that it's pretty constant all the way across. And that obviously that wasn't significant in the models, but I wanted to put that down there as a comparison. When we start to look at our 14-month PAP, you see a steady increase from 1 to 2 to 3, right? So as heart scores increase, PAP is increasing. But when we go from 3 to 4, that's a big leap in terms of the average PAP for a heart score of 4. So now here we've got marbling. So marbling, that's almost a steady linear decrease from a heart score of 1 to a heart score of 4. And as we move down on this graph, we just see that marbling is decreasing. And there's a bunch of reasons why, you can explain the way that is. When we get to the genetic correlations, this relationship is almost non-existent. So I think I have some explanations on why that is. Even though we're seeing this phenotypically, we're not seeing it from a genetic standpoint. And it's really, I think, is these animals over here are starting to, essentially fall off, and so that marbling's just not being deposited at a rate is what you would expect in a healthy animal given a fixed, you know, feeding time. And then carcass weight, and why I said that carcass weight was higher, if we look at the average carcass weight was higher, it's really a curvilinear relationship. So in terms of a hard score of 1 to 2 to 3, the heaviest carcasses we had were that hard scores of 3. And then once they go from that 3 to 4, there's that big fall off. So it's a, you know, there's that nonlinear relationship there. And I think what's happening is if you look at this, these animals are, you know, these animals are just growing and producing, growing and producing. They get to a point and then their body just falls over, and, you know, they're suffering at that point. So whether that's, you know, what are the causes of that? Not really sure, but it's just so it's really, I don't think it's the carcass weight is causing the PAP at this instance. I think that not the PAP, the heart score. I think that the heart score is causing this fall off in production. And then we have weight gain. So this is another slide. So this was weight gain across the feeding period. So, and I don't say that, I don't mean the whole feeding period. This is, so we had a subset of animals go through the grow-safe system. So, so where we got intake and everything. So in terms of their total gain in that feeding period. But what you're seeing here is as you know, heart scores of 1, it's increasing up, and it's following that similar trend for carcass weight, whereas they fall off. Yeah, they're sort of falling off that ledge there as they go from a heart score of 3 to 4. So, you know, we're seeing this rise in production and then a drop off. So, you know, whether that's it's the rise in production is causing the hypoxic environment that's causing the failing heart, and then all of a sudden they hit a point and then everything just falls off behind. I don't think we really have a good handle on that just yet. Let me skip over this slide because it's a little hard to view with all those different colors, and those colors don't show up very well. But these are the phenotypic correlations above and below the phenotypic correlations above the diagonal, Spearman correlations below the diagonal. But you'll see the biggest correlations here are with PAP and heart score, which you would expect. You know, we saw the biggest, you know, we see a phenotypic correlation of marbling, which is an antagonistic correlation. So that negative correlation, meaning as heart scores are increasing, marbling scores decreasing. We see a very small relationship between hot carcass weight. I would think this is due to that sort of nonlinear relationship between carcass weight and heart score. If that was more of a linear relationship, we would see stronger correlations here. And then again, we had another one over here with weaning weight, average daily gain and intake. I think we would see all those same sort of non-linear trends with these weight traits as well. Okay, so let's look at the sequential PAPs, because I think this paints an interesting picture of what's going on. So we have a group of cattle, as I said earlier, we have a group of cattle that have 2 PAPs, one as they entered, you know, within a couple of weeks of entering the feed yard, and then one within a couple of weeks of them leaving the feed yard. Okay, so we're calling that the 9 and 14 month. So what we've done here is we've just plotted on the x-axis the early PAP, so the 9-month PAP. We plotted the late PAP or the 14-month PAP, and we just fit a simple linear regression line in there because that's something, you know, we like to do and look at nice pretty lines. It gives us a different color, a nice blue here to look at. Anyhow, so the correlation between the 2 measures was 0.2. And this had a slope of 0.76, so meaning that for every unit increase in PAP, we would expect a 0.7 in early, every unit increase in early PAP, we would see a 0.76 increase in late PAP. But that's, to me, that's not really telling, right? So if we look at the distributions of the 2, right? So we've got histograms here of of 9-month PAP versus 14-month PAP. And you see we have a really tight distribution down here on the 9-month PAP, ranging from just above, so ranging between 30 and 50, right? And so according to Tim, that would be a pretty good PAP measures. And I think when we PAP these were all that were kind of excited that they were all kind of the same, right? So when we came back in December and did the same thing, we can't let Tim PAP when it's not freezing cold outside. So we got to bring him back in December. Can't keep him when it's just in August. But when we came back in December, we've got what you see here is this distribution's really starting to shift and spread out, right? 5 minutes. Oh, okay. All right, so let's just jump ahead here real quick as we go into just the distributions. So you see pretty much no difference between the 9 months healthy versus unhealthy, but you can see these 14-month PAPs are really starting to increase and spread out. If we look at that by individual heart score, this really drives home Tim's point as how PAP is increasing. It's really sort of destroying those hearts, right? And so you see this big exponential growth. And to me, this is really telling that, you've got a group that, of sort of hyper responders over here, in the 3s and 4s that are just, their PAP or their heart scores and PAPs are just blowing up. And then you have a group of, sort of animals that kind of tolerate, all the weight gains and changes and everything else that are going on. Well, let's skip this one because I want to get into the genetics associated with this real quick. All right, so just real quick, we've got, so we don't have a whole lot of dam information on these. So most of this is all done with the use of a sire model. And then, so we accounted for age at harvest, sex and harvest date. And that kind of, that basically accounted for their contemporary group. And then you notice we've got a couple more, I don't know why that number is a little bit off, but anyhow, it's pretty much an equal representation of bulls and heifers. So now the exciting part, I guess, is the heritability heart score is 0.28. Okay, that's what we've calculated, and I think that's pretty similar to what Justin's going to present next. Well, pretty close. So that fits right in with all of our PAP measures of the heritability for PAP. So this is the 14-month PAP, not the 9-month PAP that we've measured here, and so which is why you see a 0.94 genetic correlation between the two, essentially stating it's almost the exact same, but. This PAP heritability, if you look at literature on all the yearling PAP studies that we've done, this sits right in with all of those heritabilities that we've done for yearling PAP. And we were really ecstatic to see this heritability of 0.28 with regard to heart scoring. Now, if we look at, I'm just going to draw your attention over here to the far side real quick. So we look at the genetic correlations between heart score and back fat ribeye area and marbling. So remember, we had a pretty good relationship with marbling phenotypically, but we have a very low genetic correlation. So there's a lot of sort of other environmental noise that I think are influencing that. That's really not due to genetics or additive genetics per se. The one thing that we're seeing here is this genetic correlation between ribeye area. That's actually a moderate genetic correlation. 0.27. So where we didn't really see a phenotypic relationship between heart score and ribeye area, and really the biggest one here is this 0.63 with carcass weight, right? To me, that's really stand out on what we're seeing in terms of that relationship. That's a really high genetic correlation. So, really what this is saying is animals that have heavier carcasses are going to tend to have higher heart scores. So, if you back that up into, so what's causing the heart score, right? It's not the carcass weight, right? We talked earlier that heart score has a relationship with PAP. And if you look at the PAP in this, we see a 0.66. So it's pretty I think, telling here that that there's really something there. And if you think about this ribeye area now, you know, animals with heavier ribeye areas would tend to have, or larger ribeye areas would tend to have heavier carcasses as well. So I think that drives home some of the things we're saying. But anyhow, that's where we are. I don't know how much time. Are we done? You want? Or okay, we're done. But anyhow, if there's time for a couple questions, quick questions.
I'm trying to save a little time at the end so we can get there's questions. So,
Cody.
Was that right before slaughter, or was that earlier?
That was early on. So they were, well, it kind of depends on the group in the year, right? Because, you know, so they staggered some of those throughout, but it wasn't right up to the end. It was either early or mid feeding period.
any more?
Uh-oh.
The correlations provide, right, we cannot in workforce, does that mean I can just use that, you need to select for?
Yeah, that's a great question. So that's 14-month cut, right?
Yeah.
And so that's pat measured on the individual animal. We don't have any, you know, other pats. And so that's one of our big questions is how useful is that yearling pat, right? So if we go back and we look at the 9-month pat, there's not a whole lot of relationship there. So is it, you know, if we go to a year of age, I mean, even if we pat, you know, a wean and we pat at yearling, there's a big difference between weaning and yearling pats in our populations. So
Dr. Holt should pat at 14 months instead of 12 months.
Dr. Polk should pat at 9, 12, and 14.
9, 10, 11, 12.
But that's one of the things that we're trying to figure out how we can use that, because, you know, if we already have something out there that's useful for this, right? Or that would even add information, you know, why not use it? To me, it's you know, it would just be, it makes sense.
Quickly, Kevin.
So with the 0.28 genetic correlation on Angus influence, what about other breeds, F1s, Charolais?
I'm gonna leave that to our next speaker, because they have more of a multi-breed population to kind of flesh that out for you. We don't really have that. We're working towards that in this, but before we haven't had all the genotypes yet, so we're moving that way, and they're a little bit further on in that to answer your question. Okay,
And we can bring that back up.
we can bring that back up.
Get Dr. Holt's input. Let's thank Dr. Speidel.